So, hey guys, this is Shang Yuan from the lab of Benevento, and I'm very proud to give you this presentation about this journal in Nature about a brainstem-hypothalamus neurocircuit reduces feeling upon exposure. In brief, in this paper, Benevento and his colleagues defines a multimodal neural circuit in which tenocytes link parabrachial sensory relay to the long-time enforcement of a metabolic code, suggesting that the tenocytes constitute a novel circuital node to mediate defensive metabolic responses upon acute heat exposure. So before I will show the figures of the journal, I would like to give some backgrounds of how this research has come to place. First, the authors come to this idea by feeling that heat exposure will reduce fuel intake. For example, in summer we may not feel like the urge to eat so much. So they would like to see how our body will respond to heat exposure to reduce our fuel intake. The background also includes that the PBN region of our brain is a primary center for temperature sensing and contributes to optimizing core body temperature by increasing vasodilatation and reducing physical activity, energy expenditure, and fuel intake. The other thing to note is that the PBN region communicates with hypothalamic areas. So this study mainly cast its light on PBN region and the hypothalamic area. In addition to that, there is a cell named tenocytes which resides on the third ventricle, and these kind of cells are polarized, and it forms a cellular interface between the parenchyma and the ventricular and the vascular system of the brain by delimiting the wall of the third ventricle. The soma of the tenocytes has indirect contact with the cerebrospinal fluid, and their single basal process could extend far into the hypothalamic parenchyma, terminated around neurons or blood vessels. So, according to previous research, the tenocytes could communicate with neurons, for example, pontine neurons, that has the ability to control energy expenditure, food intake, and other stuff. So the authors here wonder that whether the tenocytes are regulated indirectly or directly to release any specific molecules to exert systematic commands to our body to control the food intake. And first, I will show you the conclusions about this journal. In this journal, the authors find that the temperature-responsive glutamatergic neurons of the PBN could innervate tenocytes, and their action potential-dependent synaptic entrainment would induce the vesicular exocytosis of VEGF-A along their basal process directly onto FLT1 positive dopaminergic and AGRP positive neurons of the arcuate nucleus, which we named the ARC. And the tenocytes could translate extra-hypothalamic sensory modalities into chemical codes to reset the output of hypothalamic neurocircuits through a direct communication with neurons. So let's see how they finish this study. First, they recorded the food intake of both gender of mice in two temperatures, the 25 centigrade and the 40. So the latter one is treated upon heat exposure. And I find that for both gender, see in figure 1b and figure supplementary figure 1a, both genders of mice have a reduced food intake after exposure to heat, especially in 2 hours. And in figure D, E, F, and supplementary figure E, they showed that they use C-force to indicate the activity of the neurons, and they find that after the heat exposure, the C-force expression in both the PBN and the vimentin plus tenocytes is induced, and the C-force expression is upregulated especially in the alpha tenocytes. So in figure 2, they try to find how and whether the parabrachial neurons innervate tenocytes. First, they find that the synaptoid contacts along tenocytes are rich in vesicular glutamate transporter 2, and this is a small vesicles that could carry the chemical signal to the neurons. They reach their approach by injecting the RAAV8 carrying M-cherry to the LV region of the brain, but they find that this transsynaptic labeling shows that the tenocytes as well as the hypothalamic neurons in the arc are marked by the M-cherry, even when they inject the virus to another region of the brain. And this indicates that the arc neurons, although they are not in the same position in the third ventricle, they might directly innervate the tenocytes. So to test whether extra-hypothalamic nuclei also innervate tenocytes, they inject the vector in the arc area, and they find that this injection can induce significant labeling of GFP in the PBN region, and the subset of PBN neurons are combined after tenocyte-driven labeling by this Cree. This indicates that some of the extra-hypothalamic nuclei also could innervate tenocytes. Then they injected a virus carrying the designer receptors exclusively activated by designer drugs to the PBN region of the mice. And in this experiment, they used C-force as an indicator for the neural activity. And they find that various neurons relay the thermal stimuli to hypothalamus. And they've tried to find out whether tenocytes are activated when exciting the PBN. So they used this clozapine nitrogen oxide to treat the PBN region of the brain, and find that this drug could significantly upregulate the C-force expression in both the M-cherry plus neurons, which was injected, and the PBN in the PBN and the alpha tenocytes. And this indicates that the PBN neurons may have some neuronal contact with the alpha tenocytes. And then they checked whether the tenocytes could respond to synaptic excitation. They reconstruct the vimentin plus processes of tenocytes to the VGLT plus terminals. And they show that more than 50 plus of the tenocytes will receive 2 or 3 the VGLT plus terminals, and the alpha tenocytes would receive more compared with the beta tenocytes. And this indicates that the tenocytes are synaptically modulated because they have to rely on this VGLT to carry their neuronal signal. Then they checked whether tenocytes respond to synaptic excitation. They used histochemical approaches to find that the AMPA receptor subunits were found in the tenocytes. And between those receptor subunits, they find GluA1 in the alpha tenocytes and the GluA2 in both alpha and beta tenocytes. The VALUT2 plus presynapses juxtaposed GluA2 plus stretches in the basal processes of alpha tenocytes. This indicates that both the alpha and beta tenocytes may rely on the AMPA receptor to carry their to respond to the synaptic excitation. And after that, they try to use a more direct way to find whether the tenocytes could respond to the excitation. They keep in this ex vivo experiment, they keep the tenocytes to a holding potential of minus 70 microvoltage, and they find that both populations of tenocytes, the alpha and the beta, has spontaneous excitatory currents upon this potential, and they find that the drug SAMPA could evoke a negative toxic current that return to baseline upon washout of this drug. So this indicates that the tenocytes could respond to the AMPA. So they also try to apply the action potential to the neurons that reside near the tenocytes, and the arrowhead shows that the tenocytes also has a calcium 2 plus triggered by the action potential. So the post hoc reconstruction of biocytin field neurons confirm that their axons indeed innervates the tenocytes. And after that, they try to find that how the PBN region can take its input to the tenocytes. They inject the vector to the PBN in the VGLT2 plus neurons, trying to induce M-cherry axons near the third ventricle and directly contact the tenocytes. And they find that the subsequent exposure to the 470 nanometer light impulses could induce excitatory postsynaptic currents in tenocytes. And this indicates that the glutamatergic neurons of the PBN directly innervate the tenocytes. After that, they try to show that the VEGFA production is changed upon the thermal challenge. So they try to screen the VEGFA mRNA expression in the alpha tenocytes, and they found that in both genders, after heat exposure, the VEGFA expression is significantly upregulated, indicating that the VEGFA production may be crucial for the response to the thermal challenge. So, they find that in the arc, both the TH plus and the HRP plus neurons could express FLT1, which is the primary VEGFA receptor. So they indicate that the VEGFA release could modulate orexigenic neurons in the arc. Then they use a VEGF receptor antagonist to see whether the spike threshold is changed after high temperature, and they find that this antagonist could occlude the change in action potential threshold upon exposure to heat. So this study can verify that the VEGFA could be crucial for the body's response to heat exposure. And finally, they try to inhibit the VEGFA to rescue the food intake. First, they try to do RNA interference to see that whether the food intake is reduced upon heat explosion. And they find that after the interference of VEGFA, the heat-induced reduction has almost disappeared. And this indicates that the VEGF-A expression is crucial for the fuel intake adjustment upon heat exposure. And then they try to inject a vector to suppress the VEGF-A production in the tenocytes. The flex gene is used to inhibit any chemical output from the tenocytes. And they find that upon the injection, the fuel intake also doesn't change upon heat exposure, indicating that the VEGF-A released from the tenocytes is required to suppress fuel intake upon acute thermal manipulation. And then they try to block the VAMP2 mediated presynaptic neurotransmitter release in PBN neurons, but not, and they see that this blocking did not influence C-force expression in them after exposure to heat, but blocking in tenocytes would cause lower C-force expression after the heat exposure, indicates that there is a reliance of hypothalamic tenocytes on excitatory PBN afferents for activation upon acute heat exposure. And after that, they try to simultaneously activate the PBN neurons and inactivate the tenocytes. But in this way, they find that the fuel intake is not changed when suppressing the tenocytes. But when they try to block the excitosis of the tenocytes, they find that the reduction led by CNO treatment is resetted. And this indicates that the tenocytes function is crucial for the controlling the fuel intake of the mice. Let's see in figure 5i, when the TLC is treated, the fuel intake of the mice cannot be reduced after the heat exposure. But when the tenocytes is not inactivated, the fuel intake is significantly reduced, as this indicates that the tenocytes is crucial for linking the PBN to feeding. So in conclusion, the neurons in the arc and the parabrachial neurons could innervate the tenocytes. And the tenocytes could respond to synaptic excitations, and after that they may release the VEGF-A to play this role in reducing fuel intake. And that is the whole of my presentation. Thank you.